To really get what we're aiming for, just take a look at the striking visual contrast we're trying to achieve here. When this protocol is done right, you get these mucin-positive glandular cells that just absolutely pop in a deep rose or magenta color, and that brilliant vivid color sits beautifully against a very pale green connective tissue stroma in the background. Plus, you've got those crisp blue hematoxylin-stained nuclei really grounding the whole field of view. That deep pink is exactly what highlights the acid mucopolysaccharide deposits so perfectly, and is what allows for incredibly precise, immediate identification under the scope.
You can really think of mucicarmine as resting on three distinct diagnostic pillars. First up is oncology. Here, it is a frontline tool used specifically to identify mucin-secreting adenocarcinomas. We're talking particularly about the colon, lung, or pancreas. The second pillar is infectious disease. In this realm, the stain is literally vital for detecting the capsule of the Cryptococcusneoformans fungus. And the third pillar is general histology, where it allows us to beautifully visualize goblet cells along with other mucin-containing epithelial cells. These three pillars are exactly what make this stain so incredibly indispensable in the lab.
Okay, before we dive down to the molecular level, we absolutely need to define the main linchpin of this entire procedure, and that is the aluminum mordant. Think of it as the essential chemical bridge that actually links the acidic mucin to the carmine dye, and it does this through a very specific electrochemical interaction. Without this aluminum mordant acting as that bridge, well, the stain simply fails. It's that important.
Now let's trace exactly how this carmine-aluminum complex works. This whole stain relies on a highly specific electrochemical interaction. Simple absorption, that won't work. Neutral mucins, they won't stand at all. The entire process is driven by the acidic charge. Over on the left, you have the acid mucin, which has these negatively charged carboxyl and sulfate groups. Because they're negative, they are magnetically drawn right to the positively charged aluminum mordant in the center. Then, this aluminum bridge connects directly over to the complex ring structure of the carmine dye on the right. Together, the three of them form a positively charged alum-carmine complex. It creates this perfect electrochemical lock, and that is exactly what allows for the targeted detection of acid mucins.
But here's the thing. All that brilliant chemistry is completely useless without rigorous pre-analytical preparation. Let's go through the checklist. First, fixation. Your gold standard here is 10% neutral buffered formalin, and you have to make sure you avoid under or over fixation. Next up, proper processing and sectioning. You want those sections cut tight, right between 3 to 5 micrometers. But here is the critical part, adhesion. You absolutely must use coated slides to prevent tissue detachment during this harsh staining process. Losing tissue at this stage? That is a diagnostic catastrophe. And finally, contamination control. You've got to maintain strict guardrails against dust, water impurities, or residual alcohol ruining your slide.
Now, equally important to your tissue preparation is human protection. We are dealing with a serious laboratory safety and reagent handling hazard matrix here. Carmine and aluminum salts are strong irritants. They require very careful handling to avoid skin, eye, or inhalation exposure. Meanwhile, hematoxylin, alcohol, and xylene bring highly flammable and neurotoxic risks right into the lab. So strict adherence to mandatory MSDS documentation, wearing full PPE, and yes, that means gloves, lab coats, and eye protection, as well as executing proper chemical waste disposal, all of that is absolutely non-negotiable.
Let's meet the players. Here is your Reagent Toolkit, featuring 5 specific chemical bottles. Every single one has a distinct, totally irreplaceable role. First, mucicarmine is your primary detector stain. Next, the 10% aluminum potassium sulfate. That acts as your chemical bridge, or mordant. Then you have hematoxylin, which provides the nuclear context, while the light green bottle offers the contrasting counterstain. And finally, your graded alcohols and xylene serve as your processing agents.
So let's actually put those reagents to work in our 7-step sequential staining protocol. Step 1 starts with deparaffinization using xylene, taking it down through alcohol and eventually into water over a span of 5 to 10 minutes. Step 2, you apply that crucial aluminum mordant for 10 to 15 minutes. Step 3 is the critical event. You apply the actual mucicarmine stain for 15 to 30 minutes. This is exactly where that primary electrochemical bonding we talked about earlier occurs. After a quick distilled water rinse, step 4 and 5 involve applying your hematoxylin and light green counterstains. Step 6, you dehydrate the tissue back through ascending alcohols to xylene. And finally, step 7, you mount the slide with resin.
When looking at the microscopic appearance, the rule of thumb is that your staining intensity correlates directly with your mucin concentration. For your positive findings, you are looking for a striking, deep red to identify those acid mucins or goblet cell granules. Or, if you're hunting for Cryptococcus, you specifically want to look for a distinct magenta capsule encapsulating the organism. Now, this brilliant red and magenta should stand out in stark contrast against your negative background. Specifically, you'll see crisp blue nuclei from the hematoxylin and a pale light green stroma from your counterstain. Let's apply this clinically. Application 1. Adenocarcinomas. As we mentioned, mucicarmine is a frontline tool for differentiating tumor origins in the colon, lung, or pancreas. It easily separates mucin-producing tumors from non-mucinous varieties. Just notice the undeniable contrast when you see it. You have these massive, bright red mucin pools that just stand out violently against the pale green stromal background. It is exactly the clear, definitive visual signal that a pathologist needs to make the call. Our second major application is infectious disease, specifically identifying fungal infections. Mucicarmine is absolutely critical for differentiating Cryptococcusneoformans from other yeast-like fungi, like Candida. If you look at the comparison, Cryptococcus presents with a central body that is surrounded by a thick, distinct, red or magenta capsule. Candida, on the other hand, lacks this mucin capsule entirely, so its appearance is very weak or completely negative. That vivid magenta halo, that is your definitive marker. To maintain strict ISO 1589 compliance and keep your clinical accreditation, standardization is just absolutely key. You're required to run a known daily positive control slide, something like colon tissue, every single day. Reagent monitoring is also a strict requirement. You must track your lot numbers and monitor the specific activity of your carmine and alum. Finally, you have to meticulously ensure thorough documentation of all your staining times and control results. This is how you safeguard against any potential misdiagnosis.
But hey, even with the strictest controls, things go wrong in the lab. So let's methodically walk through some troubleshooting for common errors. If your problem is weak mucin staining, your verified solution is to try extending the mordant time or just replacing old carmine dye altogether. If your background is overstained, you need to back off on the hematoxylin or light green time. Conversely, if your nuclei are pale, you want to increase the hematoxylin duration. Tissue detachment? That means you need to check your slide coating and handle those slides much gentler during processing. And if you're getting false positives, where neutral mucins are actually staining, you must immediately verify your reagent pH level.
Troubleshooting really comes down to a delicate balancing act, like balancing a flask on a scale. On one side, you have mordant time. Inadequate duration here prevents the dye from ever properly binding. On the other side, you have your pH level, or acidity. The pH has to be strictly maintained to ensure the stain remains specific only to acid mucins. And the fulcrum that is balancing this whole process? Dye freshness. Old carmine solution simply loses its binding affinity over time, throwing the whole scale off.
Okay, let's comprehensively lock in our final review and key takeaways. Purpose. The overarching goal of mucicarmine is to detect acid mucopolysaccharides in a brilliant red or magenta. Mechanism. It achieves this mechanically via an electrostatic bond relying on an aluminum mordant. Targets. Clinically, you are targeting adenocarcinomas, cryptococcus, and goblet cells. Controls. Operationally, you ensure accuracy by running daily positive controls. And finally, safety. You ensure human safety by strictly utilizing PPE and following MSDS guidelines.